Before birth and beyond, the enduring impact of prenatal experiences on human development. The impact of an environment on an unborn child begins long before their birth. As early as fetal development, the environment, predominantly influenced by the mother's physiological state, significantly shapes the unborn child's biological programming. The conditions experienced during pregnancy, including hormonal levels and nutrient availability in the maternal circulation, play a pivotal role in influencing the fetus's developmental trajectory. A poignant illustration of this is witnessed in the Dutch hunger winter of 1944, where the scarcity of food due to Nazi occupation led to starvation among many. Fetal exposure to this starvation during the second or third trimester resulted in a profound lesson for the body, prompting a perpetual adaptation in how it handles nutrients. Those fetuses, taught by starvation, tend to develop a highly conservative approach to storing sugar and fat, a characteristic that persists long after birth. Decades later, individuals who experience these conditions in utero are more prone to health issues such as high blood pressure, obesity, or metabolic syndrome, highlighting how the environment in utero can significantly impact an individual's health trajectory in unexpected ways. Exposing pregnant animals to stress in a laboratory setting has revealed startling outcomes in their offspring's behavior. Studies show that stressed pregnant women have offspring with a heightened inclination toward using substances like cocaine and alcohol once they reach adulthood. This stress isn't confined to laboratory scenarios. Human mothers who undergo stress during pregnancy also have discernible effects on their children. For instance, in a British study, women who experienced abuse during pregnancy exhibited elevated levels of cortisol, a stress hormone, in their placenta during birth. Consequently, their children displayed a greater vulnerability to conditions predisposing them to addictions by the age of 7 or 8, highlighting how prenatal stress lays the foundation for various mental health issues. Further research conducted in Israel focused on children born to mothers pregnant before the onset of the 1967 war unveiled alarming findings. These expecting mothers endured significant stress during pregnancy, which manifested in their offspring having a higher occurrence of schizophrenia compared to the average cohort. These profound outcomes emphasize the substantial impact of prenatal experiences on the development of an individual, underscoring the lasting influence of stress during the gestational period. Human brain development sets us apart significantly from many other species, particularly in terms of the timing and process of development. Unlike animals like horses that are capable of running soon after birth, humans are remarkably underdeveloped at the start of life. The reason behind this disparity lies in the evolutionary demands on our physiology. As humans evolved larger heads to accommodate the burgeoning forebrain that distinguishes our species, the pelvis simultaneously narrowed due to our bipedal locomotion. Consequently, human infants are born prematurely, necessitating post-birth brain development, which happens to a large extent under the influence of the environment. The notion of neural Darwinism sheds light on the brain's response to environmental stimuli during development. This concept underscores that brain circuits receiving appropriate environmental input tend to develop optimally, while those lacking such input may not fully develop or even regress. A striking example of this principle is observed in the visual circuits of a child. Even with perfectly functional eyes at birth, if the child is isolated in darkness for an extended period, the circuits responsible for vision will not receive the necessary light waves for development. Consequently, the initial rudimentary circuits, devoid of stimulus, may degrade, leading to lifelong blindness as new circuits fail to form without the requisite environmental input. This underscores the critical role of environmental stimuli in shaping and maintaining neural connections during early human development.